Hi, my name is Adam Young, and I'm a neurosurgery registrar at Addenbrooke's Hospital in Cambridge. And I'm currently doing a PhD with Robin Franklin at the Cambridge Stem Cell Institute. No, I had not worked in coronavirus before. Prior to the pandemic, I primarily worked in the immune cells of the brain, known as microglia, and how they interacted with stem cells. A significant part of my research involved the designing of viruses to use as tools in the laboratory to understand how the introduction and removal of particular genes in the brain and spinal cord would help regeneration. Well, when we were designing new viruses for different cell types within the brain, we learned an awful lot about how very subtle changes to the sequence of the virus can actually remarkably improve the efficiency for entering cells. Like many at the beginning of the coronavirus pandemic, we noticed that it was very clear that older populations were much more susceptible to the disease. What we started to look into is whether there was anything that younger children were exposed to earlier in life that may be a protective factor. So myself and Bjorn Neumann, a postdoctoral scientist in Robin Franklin's lab, started to look at the different vaccines that children were given throughout the first 5 to 10 years of life, and we analysed the sequences of these. One of the interesting things that came up from that was that actually the sequences of measles, mumps, and rubella were very similar in parts to the virus that causes COVID-19. And we hypothesized that some of these similarities that are shared could actually be predictive against poor outcome in the disease. So we originally started the analysis of the virus sequences back in March, after which, when we found some positive association, we reached out for collaboration to Dr. Yorgo Modi's at the LMB in Cambridge. Dr. Modi's then looked at how the different structures folded when the virus came together and compared these structural similarities. When we found some interesting results from this, we then progressed to look at the epidemiological data, and Dr. Alexis Ioannidis and Dr. Rocio Fernandez both did a very detailed study of different countries in Europe and looking at how there was a correlation between poor outcome in COVID-19 together with the MMR vaccine uptake. And then finally, an extra kind of clinical stage of the work was actually performed at the Luton and Dunstable University Hospital with our collaborator, Dr. Amir Riahi, who actually took some blood samples from COVID-19 patients and analysed for the presence of rubella antibodies in those patients' blood. And interestingly, what we actually found that while those patients were infected with coronavirus, it appeared that their blood cells were producing antibodies against rubella, which was obviously of great interest to our research. One of the real challenges for this project was really trying to coordinate the multiple different areas of research within the project. When the idea was originally conceived, our plan was to undertake a number of short but achievable assessments to either really disprove the hypothesis or gather evidence towards it. Trying to really organise and collaborate with everyone over a very short period from different areas was quite challenging at distance. We're really hopeful that we might actually be able to further test our hypothesis over the next few months and actually have some more conclusive evidence into the feasibility of hypothesis by towards the end of July. Of course, our project has been carried out across 3 different sites involving 4 key investigators. Even just gathering the very preliminary evidence that we have to date to really understand whether our hypothesis was viable took a great deal of collaboration. The fundamental drive behind our research is to identify if our work can provide some form of a protective intervention while a coronavirus vaccine is being created. even if our science was to prove true, the only way that there would be any real success would be if the public were to be on board and adopt the strategy. As a doctor, I know from experience that clear and open engagement with the public around vaccination is very important to build trust and compliance once new measures are introduced, and to ensure that the public concerns are accommodated and mitigated if there was to be any rollout. So currently all non-COVID-19 related research has been suspended. Generally in the Franklin group, we focus on the regeneration of the brain and spinal cord. And this is a focus mainly on diseases such as multiple sclerosis. I think if we have promising results from our original part of the project in the next few months, there certainly is a particular part of the virus which has broad relevance to our work that would be of interest moving forward. So that's something certainly as a group we would look to investigate even when the pandemic settles.